Superovulation, in vitro fertilization, embryo culture and transfer into the farm animals. Example, cow. There are four approaches in artificial breeding of animals. They are artificial insemination, embryo transfer, in vitro fertilization, and embryo cloning. Artificial insemination. As the male produces millions of sperms daily, the semen can be used to produce several offsprings. This is made possible by artificial insemination, that is AI, of females. Females are artificially inseminated here for an effective artificial insemination to produce the desired results. We have to follow some aspects. The semen ejaculate is collected. Approximately it is diluted and examined under microscope for the number of motile sperms in the sample. Normally 0.2 ml of bull semen contains about 10 million of motile sperms. The diluted semen can be used fresh within few days, or it can be cryopreserved for future use. Cryopreservation is done at -196 degrees centigrade in liquid nitrogen, as you all know, for long-term storage and transport. Artificial insemination is done on standing animals through a technique known as rectal palpation. Each semen ejaculate of a male bull can be used to inseminate as many as 500 cows. This is theoretically. Another advantage of artificial insemination is that the semen can be transported in the cryopreserved form to different places or different countries also, and used to develop superior animals. Embryo transfer. As the ruminant animal produces one egg at a time, a ruminant animal means it includes cattle, sheep, goats, horse, etc. As the ruminant animal produces one egg at a time, it can carry one pregnancy at a time. This is the known fact. It is possible to increase the production of male animals by increasing the number of mature eggs from a given female, then fertilizing them and transfer or implantation. Implant the embryos. Embryos are nothing but fertilized eggs. They are transferred into the recipient, or we say foster mother. The foster mother or recipient serves as an incubator, okay, and it does not make any genetic contribution to the offspring. Here the recipient will serve only as an incubator. It does not give any genetic contribution, or it does not pass its genetic material to the offspring. It just acts as an incubator. Embryo transfer is a costly technique. It is selectively used for the production of animals of high genetic or economic value. So what is superovulation? Superovulation can also be regarded as multiple ovulations. In the normal reproductive cycle of a non-pregnant female, one ovarian follicle, out of 20 that develops, only one ovarian follicle matures and ruptures, releasing one fertile egg at a time. The time of ovulation varies in different animals, that is 21 days for cow and horse and 16 days for sheep and goat. The circulating the circulating gonadotrophic hormone is closely associated with ovulation and release of egg. So, by increasing the concentration of this hormone, more ovarian follicles can be induced to produce more eggs. This is the concept. This process is known as superovulation, or we also call it as multiple ovulations. So here gonadotropic hormone is used to increase the chances of ovarian follicles to induce more eggs. This process is called superovulation. Superovulation may yield 10 to more than 10 or 8 to 10 eggs at a time. Some animal breeders were successful in superovulating animals to yield as many as 60 eggs at a time. This largely depends on the breed, nutrition and health of the animal. By administering prostaglandin and follicle stimulating hormone, estrus can be induced. Estrus means there will be a high fertility time for, or mating time we say, for the animals. That particular period we call it as estrus. So by administrating these two hormones, that is prostaglandin and follicle stimulating hormone, estrus can be induced. Artificial insemination is carried out in the superovulated females. Okay, after inducing estrus in the animals, in that time artificial insemination is carried out. Then it is preferred to natural mating, because artificial insemination is preferred to natural mating because the purpose of superovulation is to genetically improve the progeny, right? So instead of natural mating, artificial insemination is preferred here. As the eggs are fertilized, they undergo development to form embryos. Fertilized eggs undergo development to form embryos. From day 1 to day 4, embryos are developed into 2-celled, 8-celled morula stage and finally blastocyte stage. Another technique we have multiple ovulation with embryo transfer. Use of superovulation with the embryo transfer technique. The process of multiple ovulations, that is superovulation, we have already described, and embryo transfer are considered together, which is referred as a technique called MOET, that is multiple ovulation with embryo transfer. The embryos developed in the superovulated animals are recovered after 6 to 8 days, as I have explained already. 6 to 8 days of insemination. In case of cattle, the recovery process is easy and can be done by using a catheter, that is tube-like instrument. For certain animals with smaller reproductive tracts, surgical procedures may be needed to expose the oviduct and recover the embryos. But usually it can be done without surgical method, that is non-surgical method, by using catheter. In the picture you can see the non-surgical method for the collection of embryos using the gravity flow system. The embryos are then transferred into a synchronized recipient, that is the estrus cycle or estrus period of one, that is donor animal, is synchronized with the recipient female. So we also call recipient female as foster mother. So the embryos are transferred to the synchronized recipient. Then alternatively the embryos can be also frozen and stored. If not using at that particular time, they can be frozen and stored for use at an appropriate time and place. So about 50 to 60 pregnancy could be achieved in cattle with transferred embryos. Thus a superovulated female may result in 5 to 6 pregnancies. In this picture, it's a flow chart of embryo transfer technique. Donor cow, 8 to 12 days post estrus or follicular wave manipulation. Then superovulation is done. 5 days after initiating superovulation, we have to carry out insemination, that is, it may be natural or artificial, but they prefer artificial insemination. Then 6 to 8 days after insemination, non-surgical recovery of embryos are done. Then we say flushing process, embryo flushing, that is a removal of or recovery of embryo non-surgically from the inseminated female. Then isolation and classification of embryos, temporary holding of fresh embryos, or they can be freezed in liquid nitrogen at minus 196 degree centigrade. In order to transfer the embryo into the recipient cow, detect natural estrus or fixed time embryo transfer. Recipient cow must be synchronized with the donor cow, I have already told you. Then fresh embryo in holding to recipient cow, embryo transfer, then we can expect the offspring. Some limitations of embryo transfer. The supply of embryo from superovulated donors is limited. Freezing and thawing of embryos requires a lot of care to keep them functionally intact. The embryos, if they are not being used freshly, they must be freezed and stored in a cryopreservative method. So in that particular method, we have to freeze it, then thaw it again. In that particular process, special attention or handling techniques have to be adopted in order to keep the embryos functionally intact. Then embryo transfer requires technical skills and it is high cost. In vitro fertilization. The limitations of embryo transfer could be successfully overcome by using in vitro fertilization. In vitro fertilization basically involves fertilization of oocytes of a female animal in the laboratory conditions under artificial conditions. This is in contrast to the natural fertilization which occurs in the uterus. In vitro fertilization is reasonably successful as it results in 70 to 80% of the fertilized eggs. Through IVF technology, a large number of offsprings can be produced from a single animal. Thus, a female animal which normally produces 4 to 5 offsprings in her lifetime can produce as many as 50 to 20 offsprings by employing in vitro fertilization. Stages involved in vitro fertilization. First one is oocyte recovery. During the course of estrus cycle, the ovarian follicles grow. Okay. Then they get filled with fluid and become antral follicles, or we also call it as Graafian follicles. The ovarian follicles grow and get filled with the fluid and they become antral follicles. The oocytes from these follicles can be recovered by using laparoscopic surgery. It is possible to recover more number of oocytes from superovulated donors. Next step is in vitro maturation, that is IVM of oocytes. The immature oocytes recovered in the above step when incubated in vitro will result in mature eggs or mature oocytes. The occurrence of a large number of antral follicles throughout reproductive life in the ovaries of cattle and sheep are known. It is theoretically possible to remove them by biopsy and induce oocyte maturation in vitro. This may ultimately result in the production of hundreds and thousands of eggs from a single female. However, the potential of in vitro maturation of oocytes is limited for various reasons, that is degeneration of follicles and some unknown metabolic and hormonal conditions. Next step, we have fertilization of eggs. In vitro fertilization of the eggs is carried out by using semen obtained from a superior male animal. For IVF, the eggs are carried in small droplets, or we call them as micro droplets of culture medium. Each micro droplet usually carries about 10 eggs. A dose of sperm, approximately 1 million cells per ml, is adequate for in vitro fertilization. That particular dose of sperm is allowed to penetrate into the egg. It is facilitated by supplementing the medium with certain compounds like penicillamine, epinephrine, etc. So this is about fertilization of eggs. Then we have Embryo culture stage. The in vitro fertilized embryos must be maintained in the in vitro conditions for a few days, that is about 7 days for sheep and goat and 8 days for cattle. This allows the development of embryos to blastocyte stage. Okay, that is about approximately 60% of the in vitro fertilized embryos in vitro culture can form blastocytes. Next stage is implantation of embryos or embryo transfer stage. The 7 or 8-day old embryos from the in vitro culture are implanted or transferred in the reproductive tract of the recipient female, which acts as a foster mother or surrogate mother. Surrogate mother means, I had already told, like it doesn't pass any genetic material or genetic qualities into the offspring. It just acts as an incubator. The egg donor will be the donor cow, and the sperm will be taken from the donor animal, and they are fertilized in vitro, and that fertilized embryo is transferred to the foster mother or surrogate mother. It doesn't pass any genetic information into the offspring, it just acts as an incubator. So you can just see the steps involved here. Donor cow, oocyte is collected from superovulated donor cow. Then semen sample is collected from donor bull. Then the sperm and the oocyte, which are collected from the donors, are fertilized in vitro by specific medium. Then we can see in vitro culture, that is development of embryos into the blastocyte stage. Then the fertilized embryos can be used then freshly, or they can be freezed for future use and storage. The freezed embryos can be then thawed and used when required, and the embryos are transferred into the or implanted into the recipient cow, that is foster cow or surrogate animal. After embryo transfer, we can expect the offspring after delivery. So some of the limitations of IVF you have already know while studying about IVF in humans. The pregnancy loss of IVF embryos, particularly during the first 2 months, is very high. Although the reasons are not clearly known, some parameters are considered as the limitations of in vitro fertilization, which contributes to the fetal losses of IVF embryos, such as genetic defects in oocytes, genetic defects in fertilizing sperms, some environmental mutagenesis, that is any genetical change in the oocyte, inadequate supply of nutrients and hormones, exposure to toxic agents and free radicals. Last method of animal breeding is embryo cloning. A clone represents a population of cells or organisms derived from a single ancestor cell. Cloning basically means the production of identical copies of an individual. it would be advantageous to increase the number of embryos from a particular embryo which possesses the desired characters. Okay. There are two approaches in embryo cloning, that is nuclear transfer and use of embryonic cells. Thank you.